So the three PhD students whose work I will be talking about are Theo Josephs, Riska, and Karen Adler's work. So I will move through these stories. So just to show you where we're based, just a wee bit up the road from Oxford, a couple of hours up the road. And we established a phage centre where this work has been going on. We established it a couple of years ago. So we're now known as the Becky Mayer Centre. After last year, we received some philanthropic donation, which allowed us to actually map what we wanted to do to being able to do it. So ideally, what we're doing at the moment is establishing a large, well-curated bank of phages and attempting to act as a bit of a catalyst for the UK phage community and further beyond to help with some of the translational aspects. So that's the team of people who also contributed the stories that I will be telling today. And essentially, setting up our centre coincided with an inquiry on bacteriophage work that was done by the British government. And I've just summarised the results of this inquiry here to show that the reason why we're not translating phages largely is that there's a sort of disconnect between having effective phages, being able to produce them, and ideally we would be able to, phages would be produced, we need phages to be produced under appropriate standards, which would allow a market to be able to purchase them for these claims that we want to be able to make. And this is kind of difficult because there's still a lot of ambiguity surrounding the regulatory system, which is then limiting trials. So I am largely focusing and have largely focused on the finding the effective phages part, but increasingly at the phage centre we have projects on all these different areas, and I'll circle back to some trial work at the end of my presentation. So essentially we're in this perfect catch-22, which we're trying to unblock. Anyway, so specifically within UTIs, the reason why we focused a lot on UTIs of late is just, I mean, the numbers are absolutely outstanding. 4.5 billion people in 2021 are thought to have had a urinary tract infection. So it's large amounts of morbidity and you know, a lot of discomfort, illness, and also many sepsis cases start off as a urinary tract infection. So there's a major reason to study it. It's up to about a quarter of all bloodstream infections are thought to actually start off as a urinary tract infection. So if we look at just this cheery figure of deaths per year in the UK, you can see that actually bloodstream infections are the highest in terms of number of deaths, the number of thousands of deaths that we see every year. So being able to actually tackle urinary tract infections would have a major implication to health. And one of the things we've been trying to do of late is actually increase the diversity of phages that we capture. So when we're testing phages in therapeutic ways, we're actually able to have captured the largest diversity of phages possible. So we get the ecology generally of phages with different ecologies, because very often we isolate a few phages and then we see how effective they are, but are we actually capturing the full scale of phages that work in different ecological ways? So one of the methods that's been really led in our group, largely by Andy, but it's going to be, I think, our biggest lab effort paper. I think nearly everybody in the lab has contributed to this paper. But essentially what we've done is develop a method where we can now go from direct isolation to phage sequence within about a week for about, I think Andy's got it down to less than £10 per phage sequence. So this method is now, we sent our protocols to, they've been validated in Warwick and Sheffield, and the paper will be coming out imminently that shows how we do this. So hopefully this will be a useful approach for everybody, because it means that we can actually start off with a much greater capturing of phages before we filter down into doing the therapeutic studies. So it's using nanopore technology. And at the moment we're upscaling, we're sequencing about 500 phages a week using this method. So normally before, you would have taken your phage blacks, and then you would have purified them, and then you would have done all the work, and then you sequence them. So this is now sort of flipping that dynamic a little bit. So it's quite mad. The logistical side of it is quite mad, but hopefully it is going to be useful in lots of ways. So one of the questions I most often get in my grant applications is, well, why do you need so many phages? Like, surely you've got plenty. That's always the first thing that the referee number 2 says. And my counter argument is, yes, but not all phages evolved evenly. So in the bladders of that we just heard about in that last night's presentation, in the bladders of dogs and cats and all lots of other animals, or all the different niches that that are present for bacteria. Bacteria have evolved differently, and the phage relationships evolve differently as well. So if you're looking for phages in sort of blood, urine, lungs, they're going to have different properties, same as skin. So I've just this is just a schematic to illustrate this point, so you can see all the different types of environments that you might need. So if something's working in the bladder, which we're particularly focusing on, it's got to work in the bladder of the correct pH of the bladder. It's got to be stable in salts, and ideally, probably you would want to use phages that are stable and effective with the existing treatment. So having this bank of phages initially is going to allow you to actually find ones that have already evolved the correct properties that are the most useful. So I think these ecological insights are not really considered as much as they could be. And I'm going to just quickly mention the framework that we've been looking at in recent years. And this is the work from Theo Joseph's PhD. And largely, what we're trying to do is create some kind of ecological concept. So at the moment, we look at some things, don't we, like as specialists and generalists, but we don't have any concept of which phages, which types of, what are the properties that allow phages to survive in different environments. So we know with plants that all plants can be divided up according to 3 different properties. So this was an idea put forward by Grimes in the 1960s, and he defended it right up until the end of his research career. And it works really, really well. Plants can be categorized according to how competitive they are, how good they are at coping with stress, and how good they are at coping with disturbance. So I was interested to see if we could apply this logic to bacteriophages. And it's probably easiest to show you here. The approach that we've taken is to say, well, actually, if you think about it, when a phage infects, it has to interact with its host systems. So this is a little bit analogous to having a competition, because it competes with the host to take over it. So if a phage invests a huge amount of proportion in that early stage of transcription, that's a little bit like a competitive phage. If a phage invests a huge amount of energy in the middle, that's like a sort of stress plant, because the phage can go into the bacteria and then just kind of hunker down and survive the sort of stresses associated with the bacteria until it gets going. So that could be analogous to having a stress lifestyle, ecological lifestyle. And the final one would be if it had a lot of genes later on, that would be like something that can just immediately get to that endpoint quite quickly. So by looking at the proportion of genes at the different transcriptional stages, we could actually get some novel insights into that ecology of the host. So we took our best phages that we had discovered empirically for looking at E. coli that were causing urinary tract infections. So we had 4 really, really good phages that we'd worked out were very effective. So we looked at their transcriptional strategies. So 3 of the 4 phages were T4-like phages, and they have this as a time point along sort of early, middle, and late. And what I really want you to see here is that there's big blocks of red in the 3 different phases. So the T4 type phages appear to have this quite even. They invest equal amounts of genes are expressed most highly at the early and at the middle and at the late stage of infection. Whereas if we look at another phage, this is another highly effective phage, we can see that it actually has a different strategy. It appears to express most of its genes in this early stage. So we don't really know that. This is just sort of the very beginning, really. It was 3 years worth of quite careful RNA-seq analysis on 4 different phages at all the relevant time points. So it's a huge amount of work that Theo did. But I think it's really interesting, actually. If you look at I don't really want you can't really see this in particularly any detail, but it's more to make the point that if we look at the genes that are expressed really early, most of them are hypothetical, so we don't really know what they are. So there's huge amounts to dig out there in general. And then if we look at the host responses, if you look at the amount of overall expression of the phage and the host, we can see that the T4 type phages, this is the overall expression of the phage, and then that correlates with the closing down of bacterial transcription. Whereas if we look at the response to the phages that have this this phage had this very high middle gene expression. The host is just, you can see it's not really closed down that host transcription. The host is still fairly active. So I think digging into these relationships is going to be really interesting. And then on my triangle, this is the phage version of that triangle, that plant triangle. Many of the phages here were in this position. Here I've mapped those. So there are 42 phages that had RNA-seq done or their transcriptional profiles done. Rob's phages, for example, is done a bit with the Pseudomonas phages. So his phages are in different places in this triangle. Ours are many of them are similar to T4, but other ones are more in that stress area. So I think this perhaps gives us a new way in trying to understand which types of phages will give us the most effective results. Then we've been looking, this is now Karen's work, looking at, well, do phages actually work in ecologically relevant environments? So do they actually work in catheters? And I really like these studies. We just literally went to the clinic, grabbed the catheters from people that were being thrown, when they have their catheters were exchanged, they're being thrown away. So after about a 6-month process to get the ethical framework to do it, I should say. And when we then run our phages through an artificial bladder, we can see really clearly, this is using some micro CT scanning of the actual catheters. And we can see that before we treated with phages, you can see this biofilm. So this is treating with a cocktail of bacteriophages that target E. coli. And I think you can probably see it most clearly on this image here. Can you see that so this is an unstained image, and I really like it because it's non-destructive and all we did was literally take the catheter, show they had E. coli in them, and run our phages through. And we can see that the phages have been really effective at getting rid of this biofilm that's existing in the catheter. So I do think, following on from that talk, that it's a really good way to probably prevent infection or to treat. Now, we've also been looking at phage antibiotic synergies on a large scale. So what we did for this work was we took the sensor titer plates that they use in hospitals to see what the MICs are for antibiotics. So we used the one for UTIs, 19 antibiotics, and we tested about 30 phages and about, I think, 40 strains of bacteria. And we looked to see how effective the phages were at working in synergy with a lot of antibiotics. So people often do this with some, a smaller subset of antibiotics, but because the plates have already been made, it's actually quite a nice system to be able to overlay the phages and look at synergy on a large scale. And you can see I've just pulled out a few highlights from this analysis here. And we can see that this is a strain of E. coli that can grow really well, even at high levels of nitrofurantoin. Some of the phages didn't make any difference, but these phages have completely cleared the growth of that bacteria. Interestingly here, you can see different phages actually were effective in this case, and they're able to reduce growth in ampicillin when it was combined. But then when we added ampicillin and sulfbactam, which are used together in the presence of particular phages, we could see that the growth was prevented completely. And when we looked at, so there's a huge amount of data, like 100,000 data points that Karen's been crunching through. So this paper is about 3/4 of the way written. But what's really, really interesting is that when you look at all of these very, very large combination data sets, we can see that specific beta-lactams with specific, so this is all our antibiotics along the x-axis, these are all our phages down here, and we can see that specific phylogenetically related phages, when combined with specific beta-lactams with certain strains, are highly effective. So there seems to be an antibiotic component, a strain component, and a phage phylogeny component. So I don't think these relationships, unfortunately, are going to be that simple, but certainly we can see some clear synergies and also some antagonisms, particularly with Klebsiella. Actually, there's a lot more antagonism with Klebsiella. So the last little data set I want to show you is just the results of our mouse model. So we've established a mouse model where we can have a stable infection of either E. coli or Klebsiella for 7 days in the mice. We can count the bacteria and phage throughout that. We can count the number in the urine. And we can see that it's actually Klebsiella data. Can you see that just with one installation of phages, so after we infect the animals, establish the infection for 2 days, give phages, and we can see after a week the infection has cleared. And also, we always say phages will replicate at that site of infection. Well, this is very nice because we can see actually where they didn't have the bacteria, they didn't, obviously, there's not phages don't replicate, where bacteria are added, we see higher numbers of bacteriophages. So we've shown that one installation of phages into the bladder can remove infection, and they are replicating in that environment. We also looked at the histopathology within this environment, and we can see they were graded by a histopathology professor at a university who showed that, so this is a healthy bladder, and this is like start to get really, really crenulated as they get more and more infected. And if we look at the amount of inflammation, we can see it's much lowered in the animals that had bacteriophages. So just very quickly to touch on, I said I would circle back to the clinical use of such phages. And we know there's quite a lot of steer now from the government that the NIHR will be looking to, would fund such research. But one of the things that they demand is that you have a lot of public involvement within these projects. So we've been working with our local community. This is Melissa Haynes, the clinical person within our team, and she's working with a local team of women who are long-term chronic UTI sufferers, sort of talking about the premise of this work to make sure that they're involved right from the outset of trial design. So that data that we've gathered in the mice is now being translated into the trial design. So that's just a very, very brief oversight of these 3 PhD students. The final paper is you can see that the UTI works in bioRxiv, that's under review. The others are currently being drafted, but hopefully I've shown you that although it is a really exciting time to be developing phages, we can still see there's huge amounts we can learn by studying that natural biology and that ecology, and we need these well understood collections of phages to be able to do this. And I think working with our end users is really key right from the outset. So it just remains for me to thank all our team. Thank Thomas. You'll hear him talk, I think, tomorrow for all the sort of bioinformatic analysis that went behind this, and thank the funders. Thank you very much for your attention.